Hello, everyone. Thank you very much for the opportunity to present at the 5th International Conference on Veterinary Medicine and Health Science 2025. My name is Putri Isanumizan, and today I will be presenting a study titled Physiological Stress Response in Broiler Chickens, Gallus domesticus, transported via national and provincial routes, a study of body weight, hematology, and cortisol. Broiler chickens are a superior meat-producing breed that is widely raised across Indonesia. They play a key role in meeting the increasing demand for animal protein, and many people rely on broiler farming as their main livelihood. As the population grows and public awareness of nutritious food increases, the demand for chicken meat continues to rise every year. Transportation is an unavoidable part of broiler production. If not properly managed, this process can be harmful to the chickens and economically disadvantageous for farmers. During transport, broiler face multiple stressors that can affect their metabolism and overall physiological condition. In the study, we also take into account how transport timing, the day and versus night, may lead to different stress responses in broiler chickens. In this research, I utilized broiler chickens of the COP strain, which were subjected to transportation as part of the study design. The transportation route was developed using analogic software in collaboration with the Faculty of Civil Engineering at the University of Islam of Indonesia. The selected route comprised a combination of national and provincial routes. Transportation was conducted at two time points, at 10 a.m. during the day and 6 p.m. in the evening. Its journey lasts approximately 2 hours, covering a total distance of about 80 kilometers. So the chickens were weighed both before and after transportation. In addition, blood samples were collected for further hematological examination and cortisol level analysis. All data obtained were subsequently processed and analyzed using the SPSS software. So this is the result. This table shows the body weight of broiler chickens before and after transportation. We can see that both daytime and nighttime transport caused a decrease in body weight. However, the most noticeable drop occurred after nighttime transportation. In fact, the amount of weight loss at night was almost 10 times greater compared to the daytime group. The statistical analysis also confirmed a significant decrease in body weight in the nighttime group. This suggests that night transport had a stronger impact on the chicken's physiological conditions, especially in terms of stress and energy use. Next, the hematology result. This table shows the hematology result of broiler chickens during daytime transportation. Several parameters showed statistically significant changes. First, there was a significant decrease in erythrocyte. Meanwhile, the MCV, MGH, and total plasma protein all showed significant increases. Fibrinogen levels, on the other hand, significantly decreased. Reticulocyte percentage increased after transport, which may indicate a regenerative response. For white blood cells, including leukocyte, heterophil, lymphocyte, and eosinophil, all of them showed a notable decrease. Finally, the basophil count significantly increased following the daytime transport. Just like the daytime transport, nighttime transport also caused several significant changes in hematological parameters. First, we observed a marked decrease in erythrocyte, hemoglobin, and packed cell volume. In contrast, both MCV and MCH showed significant increases, indicating changes in red blood cell characteristic. The fibrinogen levels also decreased after nighttime transport. Reticulocyte count increased, which could suggest compensatory regenerative response to anemia. For white blood cells, leukocytes, heterophil lymphocyte, eosinophil, and monocyte all showed significant decrease. And finally, basophil showed a notable increase similar to what we saw in the day transport group. This slide compares the hematological result between daytime and nighttime transportation after treatment. Several parameters showed significant differences between the two groups, like erythrocyte count and hemoglobin levels were significantly lower in the night transport group. On the other hand, MCV, MCH, and MCHC showed significant increase at night, indicating macrocytic and hypochromic changes. Total plasma protein was also significantly lower after night transport. As for white blood cells, like leukocyte, heterophil, lymphocytes, and eosinophil, all increased significantly in the night group compared to the day group. So next is cortisol. From the table, we can see that cortisol levels increased in both day and night transport group. However, the increase was more pronounced in the night transport group. This suggests that nighttime transportation triggered a higher stress response in the broiler chickens compared to the daytime transport. To conclude, transportation using a combination of national and provincial routes causes a decrease in broiler body weight both during daytime and nighttime. It also affects several hematological parameters, indicating physiological stress response. Additionally, cortisol levels increased after transport in both groups, further confirming that the chickens experiences stress regardless of the time of the transport, though the impact was greater at night. This finding highlight the importance of managing transportation condition to minimize stress and maintain broiler health and performance. I would like to take this opportunity to express my gratitude to everyone who contributed to this research. First, I would like to thank the Faculty of Veterinary Medicine, Universitas Gadjah Mada, for the support through the research grant number 20. I also wish to acknowledge the Faculty of Animal Science, Universitas Gadjah Mada, for their collaboration and support during the study. So, and then special thanks go to all staff members in the Department of Veterinary Clinical Pathology, Universitas Gadjah Mada, for their guidance, assistance, and access to laboratory facilities. Lastly, I would like to express my appreciation to the Faculty of Civil Engineering, Universitas Islamic Indonesia, for their valuable support in developing the transportation route using analogic simulation software. Thank you very much.